Hi, my name is Emily Fleischli, and I am doing a video on melanocortin 1 receptor, or better known as MCR1. The location of this receptor is on the plasma membrane of melanocytes, or better known as skin pigment cells. The function of this receptor is to regulate skin pigmentation via a hormone known as melanocyte stimulating hormone, or MSH. For a mutated MCR1 receptor, we have the replacement of arginine at position 67 with cysteine, and this obviously impacts the receptor's function, and it leads to alteration in skin pigmentation. So for the structure, I wanted to show the primary, secondary, and tertiary structure of this receptor, and to represent amino acids, I used these colored beads. And I obviously used a special bead for the mutation at 67, which is labeled. And in my normal receptor, this is red, but in my mutated one, this bead is orange. And that kind of shows the mutation I was talking about, R to C. In the secondary structure, I showed the transmembrane receptor, and I used tinfoil to kind of show the difference in the 7 helical segments, one being uniform in its proper shape, and then one obviously irregular. For the tertiary structure, I used these straws and then a necklace that I cut to form the overall folded protein shape. And we have our regular protein, and then our non-functional irregular protein, which is altered and looks quite different. So the normal MCR1 function, as I kind of touched on, is a type of G protein coupled receptor located in the skin located in the plasma membrane and affects skin and hair pigmentation. When the receptor binds to the hormones I mentioned, like MSH, or another one called ACTH, adrenocorticotropic hormone, it kind of triggers a cascade inside the cell, and this basically leads to the production of melanin. And then melanin is responsible for the skin, hair, and eye color. So we can have a UV response by having MCR1 helping melanocytes to respond to UV radiation, and this obviously increases the melanin production, and that kind of acts as like a protective mechanism against UV damage. So more melanin produced, the darker the skin becomes, provides more protection from the harmful rays. But what happens if you have a mutated MCR1 function? Well, the R67C mutation involves changing that amino acid that I touched on earlier, and this impairs the function of the receptor, because if you change arginine to cysteine, you now have a thiol group, and this disrupts the structure of the protein. This can impact skin pigmentation. The receptor's activation may be compromised, and this can mean less melanin. And that can also cause a lighter pigmentation, so like a lighter colored white, maybe instead of black or brown. And our UV response, so the impaired function of MCR1 affects the UV response, because without the proper activation of this receptor, melanocytes are less able to respond to those UV rays and produce melanin. So this can make the skin more vulnerable. You can have more sun damage, sunburns, things like that. And again, with my model, I tried to show the differences between the normal MCR1 and the mutated MCR1. But obviously there's going to be limitations to this. The model scale is not accurate size-wise. I can't show atomic level details like the types of bonds. This model also doesn't represent the full protein environment, so there's not the entire cell membrane with all of its signaling molecules and proteins. I didn't show organelles. Obviously it's simplified, but maybe this simplicity can help you understand the mutation. It definitely helps me understand it kind of on a conceptual level before studying it in more depth.